Hello guys, myself Dr. Atul Nagpure and right now I am at CSIR-National Chemical Laboratory Pune, and the instrument what you are watching behind me is called as a molecular beam instrument, and with me we have Dr. Sadhu, scientist CSIR-NCL Pune. Now I humbly request Dr. Sadhu sir to introduce himself as well as just to highlight the importance of molecular beam technique. Yes sir, please go ahead.
Thank you for your introduction. So myself, Dr. Sadhu Kolekar, I did my PhD at Department of Physics, University of Pune, on the topic scanning probe microscopy. Then I came to NCL as a young scientist to work on heterogeneous catalysis, XPS and molecular beam. Then I went to EBS, University of South Florida. There I worked as a postdoc in two-dimensional material, its characterization using low temperature scanning tunneling microscopy, angle resolved photoemission spectroscopy, X-ray photoelectron spectroscopy. Then I went to linear accelerator at University of Stanford University. So there I worked beamline based angle resolved photoelectron spectroscopy to find the band structure of two-dimensional material. And then I went to National Lab. There I did spin polarized scanning tunneling microscopy on monolayer limit of two-dimensional materials. So now I joined as a scientist at NCL, and then I am going to explain a little bit about molecular beam instrument. So this is the instrument, molecular beam instrument. It's again a unique technique in surface science to do the reaction on the solid catalyst. And this is the chamber of the molecular beam, and then this is the gas mixing line. If you want to do CO oxidation, methane reduction and methane oxidation reaction on some serious zirconia catalyst or palladium single crystal, then you have to like mix the gases in this line, and then this is the source. So here we have mesh, very nice mesh, and then from this mesh molecules go inside the chamber. And in front of that molecular beam there is a catalyst. And once you open the shutter, inside the molecular beam there is shutter arrangement. If you open the shutter, so molecule impinge on the surface, a reaction happens on the surface, reactant gets converted into product, and that product and reactant is measured by the mass spectrometer. This is very nice and high resolved mass spectrometer. It will detect 99% molecules inside the chamber, and you will get nice spectra on the computer. And this is the manipulator, XYZ manipulator. You can rotate sample in X direction, Y direction and Z direction. If you can see through this window, you can see the direct sample, and then there is a shutter. You can on off this shutter, you see the shutter movement. So this is interesting. You can block the molecular beam, you can open the molecular beam with a fraction of second, and that happens, the reaction happens on the surface. So this is again interesting and good technique in surface science to do the reaction on solid catalyst. So later on we plan that we can integrate FTIR with the molecular beam. You see, this is the source of FTIR spectroscopy, and this side we have a detector. Now for some reason we removed the detector, but we can detect the in-situ reaction using FTIR spectroscopy and molecular beam. So that's for the idea. But now we plan to do some other experiment.
Yeah. Thank you, Dr. Sadhu sir, for sharing your knowledge and valuable time. Thank you very much, guys. See you.